In the quiet expanse of a research laboratory, a maze of cages line the walls, each containing a world of complex stories in the form of tiny, whiskered inhabitants. Among them, a brown-furred mouse named Felix led a life that seemed simple, yet, in truth, was scripted with far more intricacy than even he could understand. Felix was part of an experiment designed to illuminate the mysteries of genetics. His ancestors were brought from varied backgrounds, some tracing their heritage to the wild grasslands, others bred and rebred in the sterile order of laboratories. Each mouse carried with it a unique combination of genes, like pieces of a puzzle scattered across generations. Felix's destiny, like that of his kin, was shaped long before his first whiskers twitched. On the other side of the lab, Jade, a quiet and meticulous postdoctoral researcher, peered into Felix's cage. She noted his fur color, his quick reflexes, the way he explored corners with confident curiosity. All these traits, she knew, could be traced to the swirl of genes within each cell. She was particularly interested in a gene called agude, one that could paint fur in stripes or brown or gold, and sometimes make a mouse more susceptible to disease or resilient to hunger. Jade carefully clipped a minuscule portion of Felix's tail, a routine but essential step, and placed it in a tiny vial. Later, she would extract DNA, amplifying it to see, on a translucent screen, jagged bands revealing secrets written in the language of molecules. One mutation here could mean disease resistance, another a susceptibility to diabetes. In these mice, human ailments were mirrored in miniature, allowing Jade and her colleagues a glimpse into what makes us who we are. The lab conducted a series of crosses, pairings planned with the care of a chess master. Sometimes the result was expected, litters born with the right white patches on their bellies or the distinct twitch of sensitive whiskers. Other times, surprises leapt from the genetics, like a mouse with a tail curved in an unpredicted way or a color not seen in generations. Each anomaly sent the researchers back to their charts, wondering if they had overlooked an ancestor's hidden trait. Felix, for his part, lived unaware of the legacy he carried or the purpose for which he ran mazes and sniffed at treats. Yet, through him, Jade could carefully decode the tapestry of heredity, how genes could skip generations, how mixing two lines could create an unexpected new combination, how one subtle change could ripple through life in surprising ways. One evening, Jade stared at a newly arrived litter in Felix's cage and saw a mouse with fur far richer and darker than any she expected. This could only come from a rare genetic recombination, a twist that couldn't be planned, one demonstrating that while many details are mapped, nature always keeps the final surprise. Thus, through Felix and his kin, the story of mouse genetics unfolded in the quiet shuffling of paws, reflecting the same tangled, beautiful complexity written inside all living things.